So this video is just going to cover basically what nucleotides are and what their purpose is and all of that. Very obviously very important for cell bio as well. So let's get into it and let's start the discussion on nucleotides. So what are they? So nucleotides, in the most basic definition, they're nitrogen containing rings connected to ribose or deoxyribose sugar. So what the heck does that mean? So they're nitrogen containing rings. So they basically look like this. So that's what they look like. And they're either connected to a ribose or deoxyribose. Ribose is just OH. Deoxyribose is just H, meaning there's no oxygen, D. So RNA and DNA. So the main purpose and the main thing to figure out with these nucleotides is always what's going on that, what do you call it, that 2' sugar, which determines whether it's DNA or RNA. So what's the purpose of nucleotides? Again, they store energy. That's a big one. And they are meant for storage and retrieval of biological information. Most importantly, hereditary information. So that's good for you to know. So let's make a diagram of all the things that are in a sugar of nucleotides for ribose. So remember, what's ribose? Ribose means it has an OH, remember? So let's make the ring right now. So this is the ring, right? So the first one is just, carbon. It's the first prime, right? So this is number 1. And this is basically just OH, right? So this carbon right here is number 2. And since it's a ribose sugar, what do you think it's going to have? It's going to have an OH, right? Because it's a ribose. It's not deoxy where the O is gone. It's ribose where it's an OH. So the second prime, it determines whether it's DNA or RNA. So in this case, it's RNA, right? But always remember that. The second carbon determines whether it's DNA or RNA. So what does the third carbon do? What's the point of the third one? Let's see. So the third carbon of nucleotide sugar is important so you can add the next nucleotide to the chain. So remember how nucleotides are connected by phosphodiester bonds from 5' to 3' or 3' to 5'? This is where this comes in. So this links to some other nucleotide. So the 3' is very important for that. So we've moved on to that. Now we need to figure out what the 5' is. 4 or 5, it depends. So this part will have all the phosphate groups. So this will have, and what I mean by phosphate groups, and I'm going to make it look a little bit bigger and nicer in the next slide, is the phosphate groups are, what connects to the next nucleotide. So let's make another one. So let's say a sugar is deoxyribose. So it's a tiny little sugar right here. It has an H. It has an H because it's deoxy. It has an OH. It has an H. Right? So what the heck? What's going to happen in this box right here? Because this is obviously the phosphate group, right? So it's going to have a CH2. Then it's going to have an oxygen, a phosphate, an oxygen, a phosphate, an oxygen, a phosphate, so on and so forth. And the top part will be a double bond of oxygen, and the bottom part will be a single bond. So for phosphates, top is double, bottom is single, top is double, bottom is single. Always remember that. And that's just So it's a deoxy ATP. So deoxyadenosine triphosphate. So triphosphate, right? Well, right now that I made, okay, yeah, that's a triphosphate. So that's it for this one. So let's see what else we have for nucleotides. And one important thing that you need to understand for nucleotides is they give the phosphates of the nucleotides. So this right here, they give DNA and RNA that negative charge. So you know how you see it's negative oxygen on the bottom of the phosphate? That gives the DNA and RNA the negative charge. And they store the energy, and that energy, so when we say energy is stored, where is that energy stored? So that's a really important question. So the energy is stored in the phosphoanhydride bonds. So that's a very, very important thing to remember, and I'm going to write it down so it's ingrained in your brain. So phosphoanhydride bonds. So that energy, all of that energy, so the phosphates of the nucleotides, they give DNA and RNA that negative charge, but they also store that energy in the phosphoanhydride bonds. So that's very important for you to remember. And the last thing that we're going to talk about, which is it's going to come up next. So what's the last thing? The last thing is Stein wants us to know what the purines and the pyrimidines are. And you know this already. They're the uracils and thymine, adenine, guanine, cytosine. But how do you differentiate which one is the purine and which one is the pyrimidine? You know, like, it seems pretty the same. So always remember this way to remember them. It means who wants a cut of pie? So cut of pie, right? So pyrimidines is pie, right? And cut is obviously cytosine, uracil, thymine. And always remember for purines, it's pure as gold. Gold is Ag, adenosine, and guanine. There you go. So cut of pie, cytosine, uracil, thymine for pyrimidines, pure as gold is adenosine and guanine for purines because it's pure as gold. Get it? And also remember that the linkage between nucleotides is phosphodiester. It's a result of condensation reactions. And that's pretty much it for nucleotides. Okay, bye.